Hi, this is Brother Barnes, and this is an isotonic elodea slide straight out of the aquarium. This one has been soaked in salt overnight. Notice the clumping of the chloroplast. And then here is a side-by-side comparison. This one is kind of clumpy, but as you can see, the chloroplasts are relatively evenly distributed throughout the cells, and these are very visibly clumped. I think that this is 40x magnification on both of them. I think we were using the 4x objective on these. The idea is that in a regular elodea cell, that the cell membrane is hugged up on the inside of the cell wall, and you can actually see the chloroplast moving and grooving as the plant is living. Add salt or sugar, and that cell membrane pulls away from the wall, causing clumping of the chloroplasts. And so that's the effect that we're seeing with the hypertonic solution. Thank you to Gabby, Kayla, and Hunter for allowing me to use their microscope renditions of this little quick investigation. So look one more time, and you can see the cells here, and you really can't see the cells that great here, but you can definitely see the punctate, very dotted appearance of the chloroplasts as they are clumped together. Now this one, you can see the actual individual cells. If you look real close here, you can see that they're kind of long and rectangular. Isotonic, hypertonic, isotonic versus hypertonic.